Hello students and welcome to another session for practice problem set week 10 for the course Basics in Biology. My name is Ashutosh Behra and I am the teaching assistant for this course, which is instructed by Professor Vishal Trivedi from IIT Guwahati. So in this lecture series, the kind of pattern we have been following for this live TA session, we solve an exemplar set of questions from a given particular set of lectures, and then we move ahead learning all the concepts behind them. So this will be a live session as well as a recorded session. The recordings will be uploaded on YouTube, uh, and the slides will be shared with the students as well. So I want all the students who are attending this lecture as well as who are going to watch the recorded video, please pay attention. So this is a class from 3 p.m. to 5 p.m. and meanwhile I'll allow all the students who are trying to join this call. I will allow them inside this call, and we can start the lecture. Okay. So what I will do is I'll share my screen presentation I have made of 10 to 15 questions which are similar to what you will be facing in your assignments and in the final quizzes and exams, and these will be from a particular set of lecture series. So today we'll be solving lecture series 10, and week by week we have been progressing steadily. So lecture week 10 deals with nervous system and the brain, human brain, physiology of the human brain and nervous system. So we will deal with questions related to brain and nervous system today. And the last session will be week 12 session, as you all know, which is the end of this session, which will be 2 weeks later. So I request all of you guys to pay attention as well as call in your friends for these live TA sessions, because this is the only point of live interaction you will be having throughout this NPTEL course. So let us hope for a fruitful live session for this practice problem set week 10. So what I'll do is I will request all of the students to please turn off your video and mic and interact with me only through the chat box. I have joined simultaneously through my phone, and I'll be checking the comments and the chat box in response to the questions that I have been posting on the presentation. Okay, with that much of an introduction, let me share my screen with you guys and let's start this session. Sorry. Okay, so all of you are welcome here for practice problem set corresponding to week 10, lectures of week 10, Basics of Biology NOC 25BT02. This is the course code for NPTEL, and the instructor is Professor Vishal Trivedi, while my name is Ashutosh Behra and I am the teaching assistant for this course. I am a PMRF scholar at IIT Kanpur. So this will be the format of the presentation again. I will be posting 15 multiple choice questions which have one option correct, and I will ask the students who are attending the live session as well as in the video, recorded video session, to tell me what they think is the correct answer. Of the four options, you can post your answers in the chat box saying option A, B, or C, or D is the correct answer according to you. We will wait for some time or a minute or so, collect some answers from all the participants, and then I will reveal what is the correct answer to this question. Meanwhile, and after that, we will have a brief uh, interaction. We will have a brief understanding of the topic from which the question is related, thereby clearing our concepts and doubts regarding those topics. So if you have any doubt, please feel stop me in between post your comments, post your questions in the comment box, and I will try to address them as soon as possible. Okay, with that much of an introduction, let us start the session with the first question on board. Question number one: the human nervous system originates from dash layer of primordial germ layers. This is the question. The human nervous system originates from dash layer of primordial germ layers. Option A, ectodermal. Option B, mesodermal. Option C, endodermal. And option D, endomesodermal. You guys understood the question? The question is about the origin of human nervous system. And the question is about which germ layer, which of the three primordial germ layers does the human nervous system originate from? This is the question. Let's wait for a minute and please post your answers in the chat box. Let us keep this session interactive for mutual benefit. Okay, I think that's enough time for this question. The human nervous system originates from dash layer of primordial germ layers. And the correct answer to this question is option A, ectodermal. The human nervous system is ectodermal in origin. Now let us understand the answer about this question. What are the three germ layers? The primordial germ layers. We have the ectoderm, which is the external layer. We have the mesoderm, which is the middle layer. And then we have the endoderm, which is the internal layer. Now here is a flow chart showing all the kind of tissues, important kind of tissues that arise from these three germ layers. So if you see, ectoderm, which is the external layer, gives rise to the epidermis, obviously, because it is the outermost covering of the body. The epidermis arises from the ectoderm, skin cells of the epidermis, neurons of the brain, all the neurons in the brain, in the spinal cord, and in all our body arises from the ectoderm, and as well as pigment cells like melanocytes, which are pigment cells. These all arise from the ectodermal layer. Whereas there are other types of cells which come from the mesodermal layer, which is the middle layer. What type of cells? The muscle cells like skeletal muscle, smooth muscle, cardiac muscle, all of these arise from the middle mesoderm layer, as well as the connective tissues like blood and bone. Now what is the role of endoderm cells? Endodermal cells. Endodermal cells, the inner layer, internal layer, gives rise to cells like glandular cells and inner lining of the digestive system like the pancreatic cells, the alveolar cells and thyroid cells. So these kind of cells are produced from the endodermal layer, whereas neuronal tissue is of ectodermal origin. So the neural tissue develops from the ectoderm, one of the three primary germ layers in the embryonic development, and this process is called neurulation and involves the formation of neural plate, which then folds into what is known as the neural tube. So we have a neural plate and then a neural tube which is forming, and all this arises from the folding of the ectoderm. And this neural tube is precursor to the all central nervous system cells in the human body, meaning all neurons. So this is the first event that happens during the formation of neural tissue or the neural tube. Neuroectodermal tissue region of the ectoderm gets specified and committed to produce the neural tissue. Those are known as neuroectodermal tissue. Neuroectodermal tissue differentiate from the ectoderm and thicken into the neural plate. So what they form is called a thickened structure in the ectoderm, which is now known as the neural plate. The neural plate border separates the ectoderm from the neural plate. This is the neural plate border here shown. And beneath these are the mesodermal structures, which is the notochord, and here is the mesoderm. Then what happens, an important developmental event happens. The neural plate bends dorsally with two ends eventually joining at the neural plate borders, which are now referred to as the neural crest. So what happens is this neural plate region bends inwards and then at the borders come close to each other to touch each other to pinch off from the ectodermal layer. These border cells are now called neural crest cells. Then the closure of neural tube disconnects the neural crest from the epidermis. Neural crest cells differentiate to form most of the peripheral nervous system. So these kind of cells, the border cells are the neural crest cells, form the peripheral nervous system, while the neural tube gives rise to the central nervous system. The notochord degenerates and only persists as the nucleus pulposus of the intervertebral disc. Other mesoderm cells differentiate into somites, the precursor of axial skeleton and skeletal muscle. So other mesodermal structures differentiate to form the skeletal system and the muscular system, while the neural crest cells form the peripheral nervous system. The neural tube forms the central nervous system. Therefore, we say that all neural tissues are ectodermal in origin. I hope it is clear to the students. Just let me know with a yes or no in the chat box and we can move forward. Very good. Thank you. Moving forward to the second question. This is the second question on your screen. The subarachnoid space, which is filled by CSF, cerebrospinal fluid, is the space between dashed. I will read the question once again. The subarachnoid space, which is filled with CSF or cerebrospinal fluid, is a space between which two layers? Pia mater, dura mater, arachnoid, pia mater and brain, arachnoid and pia mater. These are the four options. Where is the subarachnoid space present? In between which two layers? Is it between pia mater and dura mater? Is it between the dura mater and the arachnoid? Is it between the pia mater or the brain and arachnoid and pia mater? So for this question you have to remember the structure of the meninges, and then you will be able to answer this question correctly. Let us take a minute and collect some answers and then we will move forward. Okay, I think it's enough time for this question and time for me to reveal what is the correct answer to this question. The subarachnoid space, which is filled by CSF, is the space between, and the correct answer is option D, arachnoid and pia mater. So this is the correct answer to this question. Subarachnoid space is present between arachnoid and the pia mater. Now let us understand about all the layers in the meninges. And what is the meninges? The meninges are the three protective and connective tissue membranes surrounding the brain and the spinal cord. So these are nothing but protective layer of the brain and the spinal cord, which are not neuronal in nature, they have other origin, but their function is to protect the brain and spinal cord from injury. These layers originate both from the neural crest cells, which are ectodermal in origin, as well as the mesoderm cells. So these are mesodermal and ectodermal in origin, both. Now this is the organization of the meninges. So in the lowermost tissue, if we have the brain, if we cut a cross section from the brain of the top of the head, from the on the lowest side, or on the lowest side we have the brain tissue. Then we have what is known as a thin pia mater, or a thin membrane like structure. Then we have the arachnoid matter, or a net like uh, net like layer. Between the pia mater and the arachnoid matter is the subarachnoid space. This is the space that we were talking about in the question. Then above the arachnoid matter we have dura mater, and finally we have the bone of the skull, or the bony structure. So before reaching the brain from the top, we first have to cross the bony structure, the skull, then the dura mater, then the pia, then the arachnoid matter, finally the pia mater, to reach the forebrain, to reach the neuronal tissue of the brain. Now let us understand the function and composition of the layers of the meninges. Dura mater, or known as tough mother, which translates into tough mother, is the outermost layer of the meninges. This is thick, fibrous, and protective in nature. This layer is derived from the mesoderm. And there are two sub-layers in the dura mater. Periosteal layer attached to the skull, and meningeal layer continues into the spinal cord. So these are the two different layers of the dura mater. And what does it functions? It forms dural sinuses that drain venous blood from the brain. So as we all know that the brain is a non-vasculative tissue. It means it does not have blood supply directly. There are no blood vessels present in the brain. Whereas where are they present actually? They are present in the dura mater. The dura mater forms dural sinuses that drains venous blood from the brain. Then comes the second layer, which is the arachnoid matter, or spider-like mother. Now what is the function of this layer and where is it located? This is the middle layer. This is thin and web-like membrane. It is almost looking like a spider web, derived from neural crest cells. So this is neural crest cells. This is ectodermal in origin, from the neural crest cells. Has arachnoid trabeculae, delicate strands connecting to the pia mater. So this is the web-like appearance that we saw earlier, which are known as arachnoid trabeculae. Forms the subarachnoid space where cerebrospinal fluid circulates. So this is the space of circulation for cerebrospinal fluid, as we just saw. Then comes the pia mater, or the tender mother, the inner layer. This is a thin and delicate membrane closely attached to the brain and spinal cord, derived from the neural crest, again ectodermal in origin, is highly vascularized, supplying nutrients to the central nervous system, helps form the blood brain barrier. So, pia mater is a constituent of the blood brain barrier, and pia mater harbors the blood vessels. The spaces between meningeal layers. Now, there are three spaces between these three layers. The epidural space between the dura mater and the skull. So, between the dura mater and the skull, there is the epidural space only in the spinal cord. Spinal canal is only in the spinal cord, but between the dura mater and the skull, there is epidural space. Subdural space between dura mater and arachnoid matter, potential space can fill with fluid in injury, and subarachnoid space between arachnoid and pia mater, which is filled with the cerebrospinal fluid. So these are all the spaces and all the layers of the meninges. They have distinct origin and distinct functions, but the subarachnoid space, which is where the CSF flows, is formed between the arachnoids layer, arachnoid matter, and the pia mater. Is it clear to the students? Just let me know with a yes or no or a thumbs up, and then we can move ahead. Very good. Okay. Moving on to the next question. Identify the correct order of meninges surrounding the brain. Identify the correct order of meninges surrounding the brain. Option A, brain, pia mater, arachnoid, dura mater, skull bone. Option B, brain, arachnoid, pia mater, dura mater, skull bone. Option C, brain, dura mater, arachnoid, pia mater, skull bone. And option D, brain, arachnoid, dura mater, pia mater, skull bone. So these are the four options before you, and you have to tell me which is the correct organization of the meninges around the brain. Starting from the innermost tissue layer, which is the brain, then when we follow upwards, what are the different layers that are present? This is the question. I think this should be an easy question since we discussed the meninges at length in the previous question itself. So this should be an easy one for you. Let's wait for a minute and collect some answers, and I will reveal what is the correct answer to this question after you guys let me know what you think is the correct answer. Okay, I think this is enough time for this question. Identify the correct order of meninges surrounding the brain. And the correct answer to this question is option A, brain, pia mater, dura mater, skull bone. Brain, pia mater, arachnoid, dura mater, and the skull bone. This is the correct order. All those who have answered, congratulations. And all those who couldn't get it correct, you will get a chance next time. You don't have to worry. Okay, let us again revisit the meninges. Now, this is a clear cut picture of a cross section from the top of the brain. The outermost layer is the skin. We let's go from inside to the outside as per the option. Brain, which is the innermost soft tissue made up of neurons. Then we have the pia mater, thin membranous like layer closely associated with the brain and the spinal cord. It helps in the formation of blood brain barrier. Then we have the arachnoid matter. Arachnoid is spider like with finger like projections, thereby it is called arachnoid. Then we have the dura mater. Dura mater connects the skull bone and skull bone with the inner layers of the arachnoid and the pia mater. Then we have the skull bone, then the periosteum, aponeurosis, and finally the skin on which our hair grows. So the correct order of the meninges surrounding the brain from innermost to outermost is brain, pia mater, arachnoid mater, dura mater, and skull bone. Understood? The pia mater, which is directly covering the brain, middle web-like layer is the arachnoid matter, and dura mater is a tough outermost layer, while skull bone is there for external protection. Is it clear to the students? This was an easy question since we were discussing about meninges at length. I hope many of you could get it correct. If yes, and if it's clear, then let's move on to the next question. Okay, thanks for letting me know. Let's moving on to the fourth question. Dash layer of meninges is made up of collagen fibers. This is the fourth question. Which layer of meninges it is made up of collagen fibers? Option A, pia mater. Option B, arachnoid. Option C, dura mater. Option D, space between pia mater and the dura mater. Which of these layer inside the meninges is made up of collagen? Is it pia mater? Is it arachnoid? Is it dura mater? Or is it the space between pia and the dura? Let us wait for a minute and collect some answers. Okay, I think that's enough time for this question, for answering this question. Question number 4. Dash layer of meninges is made up of collagen fibers. The correct answer to this question is option C, dura mater. Dura mater is the collagen containing layer in the meninges. The outermost layer of the meninges closest to the skull bone. Looking at the magnified view of the meninges, if we cut the brain open from the top, first we have the bone from the top, then we have osteoblast and periosteum of the skull bone. We have the meningeal artery. The first layer is the dura or the dura mater, which is constituted of collagen, fibroblast, dural fibroblast, and nerve cells. Lymphatic vessels also open into the dura, and there is vasculature. The bridging vein, dural sinuses, and subdural hematoma, bridging vein, pial vein. So there is a dural border, and then comes the arachnoid. The inner arachnoid and the subarachnoid space, arachnoid layers, spider-like layers with the trabeculae, which gives it a net-like, meshwork-like appearances. Then comes the pial layer, basement membrane and subpial collagen, subpial space and the pia, pial convergence with perivascular fibroblast, perivascular fibroblast layer, penetrating arterioles, virchow Robin space. So these are all the layers, and you can clearly see that the dura mater is made up of collagen. The collagen layer of the meninges is primarily found in the dura mater, which is the outermost and the toughest layer of the meninges. So obviously, if there is collagen, it provides strength to that layer and is arguably the toughest layer of the meninges. Here's how collagen is distributed across the meninges. Collagen in different meningeal layers. Dura mater is a dense collagen rich layer composed of dense irregular connective tissue with abundant collagen fibers. This provides strength and protection to the brain, to the meninges. That has two layers in the brain, the periosteal layer, the dura periosteal layer attached to the skull, and the meningeal layer closer to the arachnoid space. In the spinal cord, the dura mater is a single collagen rich layer, and then comes the arachnoid space, arachnoid matter. Some collagen in trabeculae contains some collagen fibers with its arachnoid trabeculae, which connect to the pia mater. Helps maintain the structure of subarachnoid space where CSF circulates. Then comes the pia mater, the innermost layer. It has minimal collagen, mainly delicate tissue, composed of loose connective tissue with very little collagen, highly vascularized, and adheres closely to the brain and spinal cord. And what is the function of the collagen in the meninges? It provides mechanical strength, especially in the dura mater. It maintains the structural integrity of the meninges as a whole and supports the blood vessels and cerebrospinal fluid circulation. So this is the function of the collagen, and it is densely present in the dura mater. Is that clear to the students? Just let me know in the chat, and we will move forward. Yes, very good. Great. Moving on to the next question. Question number 5. The human brain is divided into dash parts. This should be an easy question. The human brain is divided into dash parts. Option A, 1. Option B, 2. Option C, 3. Option D, 4. How many parts does the human brain have? 1, 2, 3, or 4? Let's take a minute and collect some answers, and I will reveal the correct answer when you have provided me with enough answers. I think that's enough time for this question. The human brain is divided into dash parts. Correct answer is three parts. Three parts of the human brain. And what are those three parts? The human brain can be divided into three parts: the forebrain, midbrain, and hindbrain. And these three parts have distinct functions. Forebrain or the prosencephalon. So, forebrain is also called prosencephalon. It consists of three subparts or three structures. It is the largest and most advanced part of the brain. The prosencephalon consists of cerebrum, which is also known as telencephalon. Cerebrum is responsible for higher cognitive functions like thinking, memory, emotions, voluntary movements, and it also constitutes the largest part of the forebrain. Then comes the thalamus which relays sensory and motor signals to the cerebral cortex. So the cerebral cortex is nothing but the outermost covering of the cerebrum, outermost layer of cells of the cerebrum, which constitute of neurons and which is responsible for all the higher cognitive functions. Then the thalamus is here to relay sensory and motor signals to the cerebral cortex. Then comes the hypothalamus, which regulates homeostasis, hormones, hunger, thirst, sleep, emotions. So, hypothalamus is a regulatory area which regulates many phenomena of the human body. Then comes the limbic system, which controls emotions, memory, including the hippocampus and the amygdala. So, all these important structures are part of the forebrain, which is the prosencephalon. Then the middle part is known as the midbrain, which is a small structure. Midbrain is also known as the mesencephalon. Midbrain forms a bridge between the forebrain and the hindbrain. It acts as a relay center for auditory and visual processing. It contains structures like tectum, which controls visual and auditory reflexes, tegmentum, which regulates movement and alertness. The tectum and tegmentum form the part of the midbrain, which is the mesencephalon. It is a bridge or a link between the forebrain and the hindbrain. Now comes the third part, which is the hindbrain, which is also called rhombencephalon, the primitive and vital control center. Cerebellum coordinates balance, posture, and fine motor movements. Pons, a relay center that connects the cerebrum to the cerebellum and the spinal cord. And medulla oblongata, which controls vital function like heartbeat, breathing, and blood pressure. So most of the voluntary actions are controlled by the cerebellum. Most of the involuntary actions are controlled by the medulla oblongata, and pons acts as a structure connecting the forebrain with the spinal cord and the cerebellum. Is it clear to the students that the human brain can be divided into three parts: the forebrain, midbrain, and the hindbrain? Prosencephalon, mesencephalon, and rhombencephalon. They have distinct functions. The forebrain is responsible for thinking, logic, decision making, emotions, memory. Midbrain acts as a relay center for sensory and auditory inputs and outputs. Hindbrain acts as a control center for voluntary and involuntary movements. Is it clear to the students? Okay. If yes, then let's move on to the next question. Question number 6. The olfactory lobes are present in dashed. Let me read the question again. The olfactory lobes are present in dashed. Option A, midbrain. Option B, forebrain. Option C, hindbrain. Option D, side brain. Where are the structures called olfactory lobes present in the brain? Is it midbrain? Is it forebrain? Is it hindbrain or the side brain? Let us take a minute or so and try to collect some answers from the participating students. Okay, I think it's enough time for this question. Olfactory lobes are present in option B, forebrain. Olfactory lobes are present in the forebrain of the humans. Now let us study a bit more about the olfactory lobes. So this is a fine structure of the forebrain of the humans. Now the cerebral cortex is divided into many areas regionally which correspond to a particular function carried out by the brain. We have the anterior most olfactory bulb here in the anterior most region. This area is called the temporal lobe. This is the frontal lobe associated with thinking. Primary motor cortex for motor functions. Central sulcus divides the primary motor cortex and the primary somatosensory cortex. Then we have the parietal lobe, the occipital lobe for vision, temporal lobe for hearing. These are the different regionalities inside the brain. There are many other small regions which are important and areas in the brain which where the neurons conduct a distinct function for the body. So olfactory bulb is present as a part of the cerebrum of the forebrain. The olfactory lobe, also called the olfactory bulb, is a part of the forebrain, prosencephalon, responsible for the smell. It is a paired structure located at the base of the frontal lobe of the brain. So it is there at the base of the frontal lobe, like right here. What is the structure of the olfactory lobe? The olfactory lobe consists of olfactory bulb, which receives signals from the nose and processes smell information, olfactory tract, which carries processed signals from the olfactory bulb to the brain. Olfactory cortex in the temporal lobe, it is interpreted and identified. The smells interprets and identifies the smells. Functions of the olfactory lobe: detects and processes odors, sends smell signals to the limbic system, linking smell to emotions and memory, plays a role in behavioral response to orders. So when a human smells, like this girl smells a rose flower, the smells or the molecules reach the olfactory sensory neuron, bind to them, which are protrusions of the olfactory bulb, signaling to the olfactory bulb present in the cerebral cortex. So olfactory bulb is present in the forebrain or the cerebrum and helps in odor perception in the human beings. Is it clear to the students? Is it clear to you? If yes, then let's move on to the next question of the session. Question number 7. Dash is also referred as the master gland in human body. Dash is also referred as the master gland in the human body. Option A, thalamus. Option B, epithalamus. Option C, metathalamus. Option D, hypothalamus. Which of these is also referred to as the master gland of the body? Is it thalamus? Is it epithalamus? Is it metathalamus or hypothalamus? Let me wait for a minute and collect some answers in the chat box, and then I'll reveal what is the correct answer. Okay, I think it's enough time for this question. Dash is also referred to as the master gland in human body. The correct answer to this question is option D, hypothalamus. Hypothalamus is also referred to as the master gland in the human body. Now, what is the hypothalamus? The hypothalamus is a small but vital part of the forebrain that controls many essential functions, including hormone regulation, homeostasis, and emotions. The hypothalamus is often referred to as the master gland because it regulates the pituitary gland, controls, which in turn controls other endocrine gland. So this is the structure of hypothalamus. This is the thalamus, and hypothalamus is just below the thalamus. Then we have the optic chiasm and the pituitary. Now, pituitary is also known as the master endocrine gland. Hypothalamus is the true master gland since it also controls the pituitary for its hormonal secretion. So, it controls many essential functions in turn, including hormone regulation, homeostasis, and emotions. So, here is the presence of hypothalamus. Hypothalamus is a part of the forebrain of the mammals. Now, what are the various functions of the hypothalamus? First and foremost, hormonal regulation or the endocrine control. The endocrine system, which is formed by all the endocrine glands, with the pituitary at the top. Endocrine glands, some of the well known examples are the pituitary gland, thyroid gland, adrenaline glands. The hypothalamus produces, releasing, and inhibiting hormones that control the pituitary gland, which affects the entire endocrine system. What are the examples of the hormones that are produced by the hypothalamus? Thyrotropin releasing hormone. So, this is a hormone which stimulates thyroid stimulating hormone from the pituitary. And then we have the corticotropin releasing hormone which stimulates adrenocorticotropic hormone for stress response. ठीक है ओटीपी में मैसेज कर दूंगा चलो हाँ थैंक यू sorry. Yeah, I was talking about the hypothalamus and the hormones produced by the hypothalamus in turn guide the pituitary to produce its hormone. Like thyrotropin releasing hormone stimulates production of thyroid stimulating hormone from the pituitary. Then the corticotropin releasing hormone which stimulates adrenocorticotropic hormone for stress response. Another important function of this important function of the hypothalamus is homeostasis. Now, in homeostasis, what does it mean? Maintaining the internal balance of the body, like regulating the body temperature, hunger, thirst, sleep wake cycle, and the blood pressure. So, hypothalamus also regulates the sleep wake cycle, the internal circadian rhythm of the body, which is encompassed by these functions like body temperature, hunger, thirst, sleep and wake cycle, and the blood pressure. Control of the autonomic nervous system. It also helps in controlling the autonomic nervous system. It regulates the heart rate, digestion, and respiratory rate by controlling the sympathetic and parasympathetic nervous system. So, hypothalamus also has the control for parasympathetic and sympathetic nervous system. Emotions and behavior works with the limbic system to influence emotion, motivation, and reward seeking behavior. So, it also affects behavior and regulation of circadian rhythms. As I said, the suprachiasmatic nucleus in the hypothalamus controls the biological clock and sleep patterns. So when do we sleep and when do we wake up is controlled by the hypothalamus. So in a true sense, hypothalamus is known as the master gland of the body. So on my left is a diagram which shows the influence of the hypothalamus directing the pituitary to secrete hormones like growth hormone, adrenocorticotropic releasing hormone, gonadotrophins, the MSH melanocyte stimulating hormone, ADH, and the oxytocin, prolactin, all these are stimulated by the pituitary, which in turn is controlled by the hypothalamus. Is it clear to the students? Just let me know with a yes or no in the chat box, and then we will move on to the next question. Great, thank you. Moving on to the next question, question number 8. The main function of myelin sheath in a neuronal cell is to dash generate signals, receive signals from neighboring neurons, provide insulation, and prevent leakage of ions or allow leakage of ions from the Schwann cells. Which of these is the main function of myelin sheath in a neuronal cell? I'm waiting for a minute to collect some answers, and then we will see what is the correct answer to this question. Okay, that's enough time for this question. What is the main function of myelin sheath in a neuronal cell? And the correct answer to this question is option C, provide insulation and prevent leakage of ions. Option C is the correct answer. Myelin sheath's main function is to provide insulation while a nerve transmission or while an action potential is traveling through the neuron. Now let us understand the different parts of a neuron and what is myelin sheath exactly. The myelin sheath is a fatty insulating layer that surrounds the axons of the neurons. It plays a crucial role in nerve signal transmission. So these fatty layers of layers of fat surrounding the nerve axon are known as myelin sheath. These are nothing but the end feet of oligodendrocytes, Oligodendrocytes form wrappings, extend their oligodendrocyte extends their processes which wrap the axon around in an inner tongue and the outer tongue fashion to create a fat rich myelin sheath. Now these myelin sheath plays a crucial role in nerve signal transmission. How? What are the key functions of myelin sheath? It increases the speed of nerve impulses. The myelin sheath enables saltatory conduction where electrical signals jump from one node of Ranvier to another, significantly speeding up nerve transmission. Myelinated fibers conduct impulses much faster than unmyelinated fibers. So presence of myelin sheath is not continuous. The wrappings of the oligodendrocyte are discontinuous, forming structures known as the nodes of Ranvier. So these are the nodes of Ranvier. Now what happens in the nodes of Ranvier? Nodes of Ranvier are the points which enable saltatory conduction, as in the electrical signal jumps from one node of Ranvier to the other. And myelinated neurons, so there are many neurons which are unmyelinated as such, but myelinated neurons conduct impulses much faster than the unmyelinated counterparts. It insulates and protects the neurons. So myelin sheath, since it is a fat rich layer, it is electrically insulated. So it acts as an electrical insulator, preventing signal loss or interference between adjacent nerve fibers. So when we imagine of a neuron, we could appreciate the fact that a neuron is never alone. In its vicinity, there are thousands of other neurons and other cells which also do transmit electrical impulses, which may interfere with the signal that is transmitted in a particular neuron. So what is the role of the myelin sheath? The myelin sheath will electrically insulate the axon, preventing signal loss or interference between adjacent nerve fibers. Myelin sheath also reduces energy consumption because myelination reduces the need for continuous ion exchange, making nerve conduction more energy efficient. So what happens is, we will just see in the next slide, that the presence of myelin sheath concentrates the sodium and potassium pumping in the node of Ranvier, and there is no need for a continuous exchange of sodium potassium pumping, making the nerve conduction more energy efficient, because sodium potassium pumping requires the ATP as an energy currency, and it also supports and maintains neuronal health. Myelin provides structural support to axons and plays a role in nerve generation after their injury. So myelin sheath also acts as a protective or insulative layer which provides a structural support to the axons and helps in nerve regeneration after injury. So this is what we were talking about, the saltatory conduction. Because of the presence of myelin sheath, at the nodes of Ranvier are concentrated the ion channels, and the charges are not spread over through the axon. By preventing ion leakage, the myelin sheath allows for a faster type of nerve impulse transmission called a saltatory conduction, where the action potential jumps from one node of the Ranvier to another because of the concentration of ion channels at the node of Ranvier. So these are the important functions of myelin sheath, and these are nothing but a fatty rich layer of cell extensions from the oligodendrocytes which protect the neuron at the axons. Is it clear to the students? Just let me know with a like or a yes or no in the chat box, and we will move forward. Great. Hello. हेलो. हाँ, OTP मैंने आपको मैसेज किया, आपको मिला था? हाँ 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 हाँ, ठीक है, थैंक यू. Okay, let's move on to the next question. Question number 9. The neurons which help in nerve conduction from central nervous system to the effector organs are dashed. The neurons which help in nerve conduction from the central nervous system to the effector organs are dashed. Option A, afferent neurons. Option B, efferent neurons. Option C, association neurons. Option D, interneurons. Which of the neurons help in transmitting messages from the CNS to the effector organs? Okay, I think it's enough time for this question. The neurons which help in nerve conduction from the central nervous system to the effector organs are option B, efferent neurons. These are the neurons which send signal from the central nervous system to the effector organs. So there are, what are the two major different types of neurons? One are the afferent neurons, which carry sensory info from receptors in skin, other organs to the central nervous system, aka sensory neurons. These are the sensory neurons carrying from the sensory organs to the central nervous system. Cell bodies are located outside of the spinal cord, while the endings are leading towards the spinal cord. These are the afferent neurons. For example, the skin. Neurons present in the skin conducting inside the spinal cord. Then we have efferent neurons, which carry motor info from the brain to the peripheral nervous system, aka motor neurons. Cell bodies are located in the ventral horn of the spinal cord. So in the peripheral neurons are the efferent neurons because they carry the messages from the central nervous system to effect in the motor neurons or in the motor, uh, for the movement of the somatic division or the skeletal muscles. Now, such neurons are called efferent neurons. Voluntary movement by skeletal muscles is carried out by efferent neurons. Their cell bodies are located in the ventral horn of the spinal cord. So their cell bodies has to be located inside the spinal cord. Otherwise, how will they transmit the messages from the CNS to the motor somatic system? And then there are also efferent neurons which signal the autonomic division, which regulates involuntary body responses like the dilation of the blood vessels, as shown here. So the efferent neurons carry the information from the central nervous system to the effector organs of the body. So again, the correct answer is efferent neurons, because efferent neurons or motor neurons carry nerve impulses from the central nervous system to the effector organs, which are the muscles, to trigger a response. Afferent neurons, which are also known as the sensory neurons, carry signals from the sensory receptors in the CNS, sensory receptors to the CNS. And there are association neurons or interneurons which connect sensory and motor neurons within the CNS but do not directly conduct signals to effector organs. Interneurons are another name for association neurons and function within the CNS for processing reflexes. So the reflex action follows a path of the afferent neuron, the interneuron quickly transmitting the reflex action through the efferent neurons. So here is the reflex action which is tested when you go to a doctor. Is a reflex hammer, atellar tendon. When you hit it, the information is first passing from the orange sensory neuron. Cell body of the sensory neuron is in the ventral side of the spinal cord, the dorsal root ganglion, which is now then connecting to the interneuron, and then the message is getting transmitted to the effector or efferent neurons and moving this hamstring muscle, biceps femoris muscle, flexor muscle, and the extensor muscle, quadriceps, to make the leg move. So efferent neurons are the effector neurons which transmit signal from the CNS to the effector organs. Is it clear to the students? If yes, then let's move on to the next question. Question number 10. The resting membrane potential is maintained by active transport of ions with the help of dash. The resting membrane potential of a cell is maintained by active transport of ions with the help of which kind of pumps? Option A, hydrogen ion pumps. Option B, magnesium ion pumps. Option C, chloride ion pumps. Option D, sodium potassium pumps. Now, which of these ions are pumped by active transport to maintain the resting membrane potential of a neuron? This can be a little tricky question. So let us take some time and collect answers. I'll quickly reveal what is the correct answer to this question after a minute. Okay, it's enough time for this question and time for me to reveal the correct answer. The resting membrane potential is maintained by active transport of ions with the help of option D, sodium potassium pumps. This is the correct answer to this question. Active transport of sodium potassium ions by the sodium potassium pumps helps in maintaining the resting membrane potential. Now, what is the resting membrane potential of a cell? The membrane potential is a compromise or weighted average of the equilibrium potential of all ions that can move across a cell membrane. So, all the ions that can move across a cell membrane, the sum total or a compromise weighted average of those ions are known as the resting membrane potential. So, if there are an equal number of sodium and potassium leakage channels, resting membrane potential is minus 10 millivolt, halfway between ENA and EK. It is plus 60 millivolt and minus 80 millivolt. So the resting membrane potential is minus 70 millivolt. So there are sodium potassium ions, majority of sodium ions outside and majority of the potassium ions inside the cell. There are leakage potassium channels, leakage sodium channels and sodium potassium channels. So there is the concentration gradient and an electronic charge gradient. The resting membrane potential is the electrical charge difference across the plasma membrane of a neuron when it is not actively sending a signal. It is typically around minus 70 millivolts in neurons. Minus 70 millivolts is the resting membrane potential in neurons. Key features of resting membrane potential: inside of the neuron is more negative, minus 70 millivolt, compared to the outside. Maintained by the ion concentration differences across the membrane, mainly involving sodium ions more outside the cell, potassium ions more inside the cell. Chloride ions and negatively charged proteins also contribute to maintaining the resting membrane potential. How is resting membrane potential membrane maintained? Sodium potassium pump, which is Na plus K plus ATPase, pumps 3 sodium out and 2 potassium in, maintaining the charge difference. But it requires ATP. Selective permeability of the membrane: more potassium channel leaks through the potassium leak channels than the sodium enters, making the inside more negative. So more of the potassium can leak through leaky channels to the outside than the sodium can enter inside the membrane, making the inside more negative. Negatively charged proteins inside the cell: large anionic proteins remain inside the cell, adding to the negative charge inside the cytoplasm of the cell. This makes the resting membrane potential of a neuron minus 70 millivolt, is negative. Is it clear to the students? Just let me know with a yes or no or a like, and then we can move ahead with the question. Okay, great. Now, question number 11. Which type of neurons are found in cochlear ganglia? This is another tricky question. Which kind of neurons are found in cochlear ganglia? Option A, unipolar. Option B, bipolar. Option C, multipolar. Option D, apolar. Which of these type of neurons are found in the cochlear ganglia? Let's take us some time and then come to the answer. Okay, I think it's time for me to reveal what is the correct answer to this question. Which type of neurons are found in cochlear ganglia? The correct answer to this question is option B, bipolar neurons. Bipolar neurons populate the cochlear ganglia. Now, what are the different kinds of neurons? Neuron can be divided into many, classified into many types depending on the morphology and the position of the cell body and axon. This is a typical bipolar neuron on my left, cell body in the middle with two axons. The dendritic zone synapses on cochlear hair cells, while axon terminal synapses in CNS. Then we have a unipolar neuron with one cell body protruded out of the two axon terminal, branched axon terminal, receptor free nerve endings and axon terminals in CNS. Then a multipolar neuron with dendrites connecting to the cell body at one end and an axon and axon terminals at the other end. So this is the structure of the inner ear of the cochlea, showing all the structures. This is the hair cells which flow from the sound information, the membrana tectoria, cells of the Claudius, cells of the Henson, interphalangeal cells of the Dieters. But what we need to focus are these neurons which form the spiral canal containing spiral ganglion neurons or the SCN neurons, which are bipolar in nature. Central processes of the bipolar neurons of the spiral ganglion forming the cochlear nerve. So these bipolar neuron emanate axons which form the part of the cochlear nerve, connecting one end to the cochlea and the other end to the auditory lobe. The cochlear ganglia neurons, also called the spiral ganglion neurons (SGNs), are specialized sensory neurons located in the cochlea of the inner ear. They play a crucial role in transmitting auditory information from the hair cells in the cochlea to the brain via the auditory nerve or the cochlear nerve, part of the cranial nerve 8, vestibulocochlear nerve. Structure and function of the cochlear ganglion neurons. They are found within the modiolus, the central axis of the cochlea, organized in a spiral shape, giving them the name the spiral ganglion neurons. They convert mechanical sound vibrations into electrical signals and transmit these signals to the auditory cortex via the cochlear nerve. What are the types of cochlear ganglion neurons? Type 1 neurons, 95%, which are large myelinated neurons, make one-to-one connection with the inner hair cells and responsible for high fidelity auditory signal transmission. And the type 2 neurons, which are few in population, 5%, unmyelinated smaller neurons, connect to multiple outer hair cells, may play a role in modulating the sound sensitivity. So these are cochlear ganglion neurons which are bipolar in nature, connecting one end to the hair cells which transmit mechanical sound vibration to electrical signals and then transmit these signals to the auditory nerve of which are the efferents, nothing but the efferents of the cochlear bipolar neurons. I hope it is clear to students. Okay, great. Moving on to the next question. Question number 12. The sympathetic nervous system belongs to dashed. The sympathetic nervous system belongs to option A, central nervous system, option B, peripheral nervous system, option C, autonomic nervous system, option D, non autonomic nervous system. These are the four options. Which nervous system does the sympathetic nervous system belongs to? Let's take a minute and collect some answers. Okay, it's enough time for this question. The sympathetic nervous system belongs to autonomic nervous system. Option C is the correct answer to this question. Autonomic nervous system. Now, the sympathetic nervous system is a part of the peripheral autonomic nervous system, which constitute of two arms, the parasympathetic nervous system and the sympathetic nervous system. Sympathetic nervous system mobilizes reserves under the stress, while parasympathetic nervous system maintains homeostasis. So, what are the key functions of these two nervous systems? Sympathetic nervous system dilates the pupils, while parasympathetic will constrict the pupil. It inhibits saliva production, the sympathetic nervous system, while parasympathetic stimulates saliva production. Sympathetic nervous system also relaxes the bronchioles, increases heart rate, slows down digestion, reduces intestinal muscles, stimulates glucose release, stimulates adrenaline production, and stimulates orgasm. While the parasympathetic nervous system does the opposite of all these sympathetic functions. It constricts the bronchi or relaxes the bronchi, slows down the heart rate, stimulates the production of bile for digestion, stimulates digestion, and stimulates sexual arousal. The sympathetic nervous system is a part of the autonomic nervous system which controls involuntary functions like the heart rate, blood pressure, and digestion. And the autonomic nervous system has two main divisions: sympathetic nervous system, fight or flight response, increases heart rate, dilates pupils, etc. Parasympathetic nervous system, rest and digest response, which slows the heart rate and increases the digestion. So one is a fight or flight response nervous system, which is sympathetic, and rest and digest is the parasympathetic nervous system. Easy to remember, right? Is there any doubt? Is it clear to the students? Yes, great. Looking at the 13th question, optic chiasma is found on the anterior surface of dash. Optic chiasma is found on the anterior surface of option A, diencephalon, option B, thalamus, option C, hypothalamus, option D, epithalamus. Where is the optic chiasma found? If you remember it correctly, if you have followed or paid attention to the earlier slides, you might be able to answer this question because we have dealt with all the structures of the nervous system, and optic chiasma is one such structure. Now you just have to recall where have you seen it or where have you heard its name. Okay, let's take a minute to get back to this question where I'll reveal the correct answer. Okay, let's get back to this question. Optic chiasma is found on the anterior surface of hypothalamus. Option C is the correct answer. Hypothalamus anterior surface contains optic chiasma. Now, what is optic chiasma? It is the X-shaped cross section of the optic nerves from the both eyes in the optic tract. This is the optic chiasm, forming a X-shaped sky-shaped structure. Hypothalamus, which regulates circadian rhythm, the anterior portion of the hypothalamus has the optic chiasm, right? So the optic nerve from the eyes crosses each other from the right to the left in the optic chiasm. Here is the pituitary gland showing the optic tract and the visual radiations transmitting it to the primary visual cortex present at the back of the head. The optic chiasma is a structure located on the anterior surface of the hypothalamus where the optic nerves, the cranial nerve 2, partially cross. Key features of the optic chiasma lies in front of the hypothalamus near the base of the brain and positioned above the pituitary gland and the infundibulum, which is the stalk connecting the hypothalamus to the pituitary. It is the point where the medial fibers of the optic nerves cross to the opposite side and the lateral fibers remain uncrossed. This crossing ensures that visual information from each eye is processed in the opposite visual cortex. So this is the catch, or this is the function of the optic chiasma, that visual information from each eye is processed in the opposite visual cortex, from the left eye to the right visual cortex and from the right eye to the left visual cortex. Lesions in the optic chiasma can cause bitemporal hemianopia, leading to loss of peripheral vision, important for binocular vision and depth perception. So optic chiasma is important for the binocular vision and the depth perception. I hope it is clear to the students. If yes, then let's move on to the next question. Question number 14. Longitudinal myelinated nerve fibers called cerebral peduncles are located on dashed. Longitudinal myelinated nerve fibers called cerebral peduncles are located on dashed. Option A, forebrain. Option B, midbrain. Option C, hindbrain. Option D, pons varolii. Which of these structures have cerebral peduncles? Let us take a minute and then get back to this question. Okay, time to come back to this question. Longitudinal myelinated nerve fibers called cerebral peduncles are located on the midbrain. Option B, midbrain. Midbrain consists of these tracts, axonal tracts, which connect the forebrain to the hindbrain, called cerebral peduncles. So this is the structure of a cerebral peduncle, the whole structure of the midbrain, and these are all the parts of the midbrain. The cerebral peduncles are a pair of large myelinated nerve fiber bundles located on the ventral surface of the midbrain. So this is on the ventral surface of the midbrain. They form a bridge between the cerebrum and the lower brain regions, the cerebellum, including the pons and the spinal cord. The cerebral peduncles are divided into crus cerebri, the anterior part, contains descending motor tracts from the cerebral cortex, corticospinal, corticobulbar, and corticopontine tracts, plays a key role in voluntary movement control. So this crus cerebri of the cerebral peduncle plays a key role in voluntary movement control. Tegmentum, which is the middle part, houses important nuclei like the red nucleus and the substantia nigra, involved in motor control, coordination, and the dopamine production. Substantia nigra is linked to Parkinson's disease. The pretectum and the tectum contains the superior and inferior colliculi, which are important for visual and auditory reflexes. And it also houses the pineal gland, the midbrain. So these are all the functions of the cerebral peduncle involved in connecting the forebrain to the lower parts of the brain, which are spinal cord and the medulla oblongata. The crus cerebri anterior part involved in voluntary movement, tegmentum which contains substantia nigra and motor control, dopamine production, while pretectum and tectum which are important for visual and auditory reflexes. I hope it is clear to the students. If yes, then let's move on to the final question of this session. Which spinal nerve has the same number of nerve pairs as that of lumbar spinal nerve? Option A, cervical. Option B, thoracic. Option C, coccygeal. Option D, sacral. Understand this question again. Which spinal nerve has the same number of nerve pairs as that of lumbar spinal nerve? Okay, it's time for me to reveal what is the correct answer. Which spinal nerve has the same number of nerve pairs as that of lumbar spinal nerve? Option D, sacral. Let us look at all the different kinds of spinal nerves present in our body. From the spinal cord emanating all these nerves are the spinal nerves. Spinal cord sections and nerve groups. First mentioned in the cyan color or the blue color, cervical nerve pairs, 8 nerve pairs. Then comes the pink labeled thoracic, 12 nerve pairs. Then comes the green labeled lumbar, 5 nerve pairs. Labeled in purple is sacral, 5 nerve pairs. And the last is the cauda equina. So from the pia mater and the arachnoid and the dura mater, these spinal nerves arise. The lumbar spinal nerves consist of 5 pairs, L1 to L5, and the sacral spinal nerves also consist of 5 pairs, S1 to S5, making them the correct match. So the question was, which spinal nerve pair have the same pairs equal to lumbar? Spinal nerve pair distribution: cervical 8 pairs, C1 to C8, thoracic T1 to T12, lumbar L1 to L5, sacral S1 to S5, and coccygeal 1 pair, CO1. If this is clear and understood, then we have come to the end of the presentation. The spinal nerves are mixed nerves, meaning they carry both the sensory and motor efferent and afferent signals between the body and the central nervous system. Cervical nerves control the movement and sensation of the neck, shoulders, arms and hands. C3 to C5 contribute to the phrenic nerve, which controls the diaphragm for breathing. It comes from the cervical nerve, not from the thoracic nerve. Thoracic nerves control the muscles for chest and upper back and help with breathing, controlling intercostal muscles and provide sensation to the torso. Lumbar nerves, on the other hand, control lower back, hips, thighs and parts of the legs, important for walking, bending and lifting. Sacral nerves control the pelvic regions, buttocks and lower legs, help regulate bladder, bowel and sexual function, while coccygeal nerves provide minor sensory function to the skin near the tailbone. With this, we have come to the end of this presentation as well as the class. I will stop my screen sharing here. Yeah, thank you all for bearing with me for 2 long hours and solving together these 15 questions corresponding to lecture week 10 for basics of biology. And those who will be watching this recorded video, you can access these slides which will be provided to you by the NPTEL authorities, while this recorded class will be currently uploaded in YouTube. You guys will be provided the link from the NPTEL authorities. So I hope the class was understood by everyone and everyone could follow, brush up their concepts. And as per every time, I would say please read your lectures, go through them, or at least attend the live sessions to have a clear grasp of this subject. It is an easy subject, and you need to dedicate some time to understand this completely. Okay, with that much, I will end this class, and let's meet next Saturday dealing with practice problem set of week 11. Thank you for joining this call, and bye bye.